Greetings everyone. Today on this happy, beautiful occasions, me and my group will be presenting our student projects entitled Genetic Codes. Before we start our presentations, please allow us to introduce our team. Please, next slide. And here is our team. You can read it yourselves and you can read it yourselves on the screen with each of our names listed there. And please, next slide. Here is our table of content. We'll be covering a few chapters here. In chapter 1, chapter 2, we'll be covering about the definitions, origin of genetic codes, genetic code characteristic, gene expression, the function of genetic codes, the mutation and disease, and also we will end the presentation with conclusions of our own. Next. And just start on the background of our student projects, and my friend will explain it for you. Please.
Okay, hello, my name is Yanti Graciella and my name number is 22025111162. So I will start by explaining the background. Sometimes when we try to compare someone with his or her surrounding, we are born to meet someone if not lots of differences or in his or her physical appearance or behavioral state. For among our peers, friends, siblings, parents, or even other family members, there will always be differences. This variety of differences applies to everyone. Even identical twins are no exception, thus making every human being unique and special as they are. The cause of all differences are actually contained inside of our body, inside our smallest component, which is a cell. Next.
All right, let me introduce myself before I explain my part. My name is Herberth. My number is 2202511169. There is the purpose of our student projects. It's to know the origins of the genetic codes and to know the functions and essentials of genetic codes, and also to know what affects our genetic codes. And as for the benefits, it's through broadening the reader's knowledge about genetic codes and related matters, improving overall health, both individually and socially, enhancing our awareness about genetic codes and other important health factors. Next.
My name is Nadira. My student number is 22025111160. I'll be explaining about the definition. So genetic code is a set of rules located in DNA and RNA that is translated by living cells to form amino acids for proteins. Genetic code is composed by codon, a nucleotide sequence that is used to make the polypeptide, scanned from 5 to 3 on the ribosome in groups of 3 nucleotides. Proteins used for synthesis are actually from the messenger RNA, not from DNA. RNA is composed of 4 different nucleotides: adenine, cytosine, guanine, and uracil. In total, there are 64 codons. 3 of them do not code for any specific amino acid called subcodon, which also indicates the termination of protein synthesis. Additionally, there is also a codon which initiates the translation stage called start codon. The start codon has AUG as its sequence and specifies methionine for the amino acids. Next.
Hello, my name is Adellya. My name number is 22025111165, and I am gonna explain the origin of genetic codes. One of the hardest problems in research on the origin of life is to find the origin of genetic codes. The development of genetic code is related to the characteristics of genetic codes. In general, there are 5 phases of genetic development. First, the beginning. At first, the genetic code was not known for its structure and it didn't produce specific protein. It was ambiguous because at a time it translates protein that another time translates another unidentical protein. Second, evolution. The development of genetic code works as a whole, not only the kind of protein it wants to produce. This evolution continues until ambiguity is removed and specific proteins produced. Third, optimization. Modern genetic code is derived from the ancestral genetic code. This optimization is used to reduce failures and mistranslation. For major transition, the prerequisite for a major transition is the complete disappearance of ambiguities. With this ambiguity removed, the genetic code synthesizes specific proteins. Fifth, conservation. The refinement of protein in the genetic code leads to some limitation of various possibilities, and its active biological mechanisms are continuously at work to evolve and to restore their original structure. Next.
Hello, my name is Putu Asweni Pururaswini Astawa. My name number is 2202-511161. I will explain about DNA characteristic. Number one is DNA characteristic is universal. It means all of the organisms have the same genetic code because there is a kinship in evolutionary history. This also means the same codon will be translated to the same amino acid. Number two is non-ambiguous. One codon that determines one type of amino acid. Codon is the genetic code carried by mRNA, which is formed for triplets in mRNA and serves to determine the type of amino acid. Number three is Comlis code. There is no punctuation between consecutive codon. The next characteristic will be explained by my friends. Thank you.
So, DNA is not comprehension. It means that the nitrogen species would make up the combination of slash, which means that a codon has a different base sequence than the unfolders. Yes, DNA is redundant. It means it can also be called integrate. One amino acid can be coded for 4 by many proteins. And then DNA is replicable. It means the codon consists of 3 constituents, 3 sequences, depending on the amino acid. The direction of the is always correct.
Hello, my name is Michelle Carolina Putri and my name number is 2202-511167 and I will explain about gene expression. Next. So, gene can be expressed in the process of using genetic information contained in genes to synthesize gene products. In general, gene product compounds are proteins. In addition, gene products are also available in the form of functional RNA compounds such as transfer RNA, ribosomal RNA, and small nuclear RNA. The process of gene expression must go through 2 stages, namely transcription and translation. Next. Transcription in the process of forming a single strand of RNA by using a DNA strand as a template. Transcription in eukaryotic cells occurs in the nucleus and takes place in the cytoplasm in prokaryotic cells. Not all parts of the DNA strands will be transcribed, but only the part that contains the gene. In the transcription process, the role of RNA polymerase enzyme is needed. In eukaryotes, there are 3 types of RNA polymerase, namely RNA polymerase 1, 2, and 3. They have different roles, whereas in prokaryotes, there is only 1 type of RNA polymerase, which plays a role in synthesizing all types of RNA. Next. The transcription process is divided into 3 stages as follows: initiation, elongation, and terminations. For further details, you can find them in the slides. After transcription process, it will be post-transcriptional modification. In eukaryote cells, the resultant RNA from the termination stage is not yet able to function. Therefore, post-transcriptional process and modification are required to form a well-functioning eukaryotic RNA. RNA processing achieves 3 things, namely adding 7-methylguanosine at the head of 5 strands, adding poly A to the tail of the 3 strands, and performing splicing by removing the introns. Then RNA transport transport from the nucleus to the cytoplasm. The process of transcription in eukaryotic cells that occurs in the nucleus. Discuss the final products of transcription, mRNA, to be carried to the cytoplasm through the nuclear envelope. In the cytoplasm, mRNA will bind to this ribosome for the next process, translation.
Hi everyone, let me introduce myself. My name is Patricia or Jotan, and my student number is 22025111171. So translation is a process where the nucleotide sequence of a mRNA is used as a template to connect with the amino acids of polypeptide chain in correct order. Protein synthesis happens in cytoplasm in eukaryotic cell. 3 types of RNA molecules come together to perform different but coordinated function. Types of RNA used in this process, the first one is mRNA or messenger RNA, conveys the genetic information transcribed from DNA in a linear form. The mRNA is read in a 3 set of nucleotide sequence called codons, each of which specifies a particular amino acid. The second one is transfer RNA or tRNA. It's the key to decoding codons in mRNA. Each amino acid type has its own subset of tRNAs covalently attached to the amino acid, which carries it to the growing end of the polypeptide chain when the next codon in the mRNA calls for it. Each specific tRNA molecule contains an anticodon, a sequence of 3 nucleotides that can base pair with the complementary codons in mRNA. So the correct tRNA with the added amino acid is selected at each step. The last one is ribosomal RNA, associates with a set of proteins to form ribosomes. This complex structure that physically move along the mRNA molecule catalyze the assembly of amino acid into polypeptide chain. They also bind tRNAs and various accessory proteins necessary for protein synthesis. Ribosome consists of a large subunit and a small subunit, each containing its own RNA molecule. Next slide, please. So the translations process divided into 3. The first one is initiation, elongation, and then the last one is termination. The complete process of each can be read at the slides on the screen. And after that, the process called post-translational modification. Newly synthesized polypeptide chains must undergo post-translational processing to produce the final protein. That's it for me. The next slides will be presented by my friend. Thank you.
Let me introduce myself. My name is Putu Ayudhea A, and my name number is 22025111164. And I will explain about the functions of the genetic codes. The function of the genetic codes could be seen in protein synthesis. Protein synthesis is a process that translates genetic codes in DNA to become visible features in organisms. This process occurs in the cell nucleus and ribosome. Protein synthesis involves adenine, cytosine, guanine, and uracil. The information of the genetic codes are determined by the nitrogen base of the DNA chain. Next.
Okay, hello everyone. My name is Evrilla Deidree and my name number is 2202511163. I will be explaining about DNA mutation. Mutation in human genes can be responsible for inherited genetic disorders, and a genetic disorder is a disease caused in whole or in part by a change in the DNA sequence away from the normal sequence. Next. Genetic disorders can be caused by 4 different things. The first one is mutation of one gene or monogenic. Mutation can happen as a result of substitution of one nucleotide with another or insertion of a nucleotide into a sequence or deletion of a nucleotide from a sequence. The second one is mutation of multiple genes called multifactorial inheritance. These consist of some physical and intelligent traits of individuals which are modified by parents and affected by nutrition and environment. The third one is mutation of one or more chromosomes. This is a mutation that affects any of the 46 chromosomes in the nucleus of a human cell. The last one is epigenetics. Epigenetics are a set of events that can alter how the body reads DNA without any change to the original DNA sequence. Next.
Hello everyone, my name is Cendrawan Han Santoso with my last 3-digit name numbers is 170. Some diseases resulting from monogenic type of mutation. The first is cystic fibrosis. It occurs when there is a mutation in the CFTR gene. And the second one is sickle cell disease. It occurs when there is a mutation in the HBV gene. And sickle cell anemia happens when there is a change in amino acid from glutamate to valine. Next. And these are also some diseases resulting from chromosomal mutation. The first is Down syndrome. It is caused by a trisomy of chromosome 21, an extra copy of chromosome which affects the development of the baby and impedes the physical and mental growth. Second is Turner syndrome, which is caused when part or all of an X chromosome is missing or abnormal. And the third is Klinefelter syndrome, condition caused by trisomy of chromosome X. It affects males. Next. For multifactorial inheritance, it is caused by 3 major factors: inheritance of genes by parents, nutrition, and environment. For epigenetics, these are the agents that drive epigenetics, which are, for some examples are virus, bacteria, hormones, and nutritions. While some of the processes that occurs in epigenetics are methylation and acetylation.
Okay, I'm Wildan Mahsyar. My number is 220-511-174. Conclusion. Genetic code is a set of rules located in DNA and RNA that is translated by living cells to form amino acids for proteins. Genetic code is composed by codon, a nucleotide sequence that is used to make polypeptides. The genetic code possesses several characteristics, which are universal code, non-ambiguous code, redundant code, triplet code, non-overlapping code, and homologous code. Genes can be expressed in the process of using genetic information contained in genes to synthesize gene products. This event is called gene expression. The process of gene expression must go through 2 stages, namely transcription and translation. The transcription process is divided into 3 stages: initiation, elongation, and termination. After transcription, post-transcriptional modification and RNA transport from nucleus to the cytoplasm occurs, and finally the translation process. Mutation in human genes can be responsible for inherited genetic disorders. Genetic disorders is caused in whole or in part by a change in the DNA sequence away from the normal sequence. Genetic disorders can be caused by mutation in one gene or monogenic disorder, mutation in multiple genes by a combination of gene mutation and environmental factors, and or by damage to the chromosome. That's all our presentation about genetic codes. Thank you very much. Stay healthy and be well.